Our next speaker is Nikki Schultz, who comes from Memorial Sloan Kettering Cancer Center. Nikki leads the development of cBioPortal, which is something that all of us need to be familiar with, so I'm very pleased that Nikki was able to fill in for Ed at the last minute. Nikki is involved in several projects in the TCGA, is also an investigator in the Stand Up to Cancer Prostate Cancer Dream Team. He's an associate attending computational oncologist in the Department of Epidemiology and Biostatistics and an affiliate of the Human Oncology and Pathogenesis Program, and he is head of knowledge systems in the Marie Yosei, did I say that right? I'm trying, and Henry R. Kravis Center for Molecular Oncology. So Nikki, tremendous thank you for filling in on just about 7 days' notice. We really appreciate it, and we need to hear your expertise. Thank you.
Thank you, Lee. Thank you for the invitation. Thanks for letting me speak here. So yeah, I'm a molecular biologist by training who's transitioned into a more computational role during my PhD and then my postdoc. And the one thing that has taught me is that there's a huge disconnect between biologists and clinicians who are challenged with interpreting these large amounts of genomic data that we get now from patients, and the computational people on the other side who have the computational skills to process all that data, but not the biology background to really interpret it. And these two fields don't really speak the same language. And so I've, over the last 10 years, tried to bridge that gap and try to build tools that are useful for clinicians and biologists to interpret the data. And I'll talk about some of those tools here today. And Louise alluded to this, when you sequence cancer samples, there are millions of variants that you can find, and the vast majority of them, we don't know what they are, we don't know what they do. We actually think that most of the mutations that we find in cancer samples are what we call passenger mutations. They're not contributing to the growth or the development of the tumor. They're just passengers that are there along for the ride. And this famous graph here by Gady Getz's group from about 5 years ago illustrates this quite nicely. What you're looking at is hundreds or thousands of tumor samples that the Broad had sequenced at the time. These are exome sequencing, meaning every exon of every gene was sequenced. And what's plotted here on the y-axis is the number of non-synonymous mutations found in each of these tumors. Each dot is one tumor. And the y-axis is on a log 10 scale, so you have orders of magnitude of difference in number of mutations between the GBM sample here at the top and then this rhabdoid tumor at the bottom. So tumors up here on the right have a much higher mutation burden. Those are the ones that tend to be driven by exogenous mutagens like the sun in melanoma, smoking and lung cancer. Some of them have endogenous mutagens like polymerase epsilon mutations in colorectal and endometrial cancer or microsatellite instability. And then the tumors on the left tend to be those that are childhood tumors that just didn't have much time to develop mutations, or hematologic tumors. But just because these tumors on the right here have orders of magnitude more mutations doesn't mean they have that many more driver variants. So our challenge in interpreting what drives a tumor is figuring out what these mutations are. Another example is shown here. Even though you think you know what the driver genes are, that doesn't mean that every variant in these genes is also a driver. And this is data aggregated from about 10,000 different tumors showing EGFR and ERBB2 mutations. You're looking at the linear domain structure of the proteins from left to right, and then the number of mutations found at each amino acid position on the y-axis shown in little lollipops. And you can see almost all amino acids in the protein have been found to be mutated at least once, sometimes twice, across large cohorts. But that doesn't mean all of them are drivers. What's pretty clear and what we begin to realize is that the more commonly mutated amino acids are those that are the ones that are drivers. So figuring out which ones they are among the sea of background mutations is a challenge. And then, of course, once you know which ones the drivers are, that still doesn't mean that you can act on those. Only a subset of the known drivers are actually clinically actionable. But you want to know which ones those are as well. So how can we identify these driver variants in cancer samples, and how can we find the actionable subsets? And I'm going to go through 3 approaches that we take. The third is really a combination of the first two. The first one is simple recurrence. If something is more frequently mutated than you would expect by chance, it's more likely to be a driver. Second one is obviously prior knowledge. If we have found something to be activating or sensitive to a drug and that's documented, we should be able to put that into a database and make use of that data. And then the third one is combining all of that in intuitive, easy-to-use visualization tools. So I'll briefly touch on a method that we helped develop called cancer hotspots that simply looks across large collections of tumor samples and tries to find not the frequently mutated genes, but the frequently mutated positions within the genes. And this is work that we did that was mostly driven by Barry Taylor's group at MSK, but we helped them put together a resource called Cancer Hotspots, which is accessible at cancerhotspots.org. And when you run this method, I won't have time to go into details. We did this across 24,500 tumor samples. You find hundreds of hotspots in dozens of genes, and just illustrating the ERB2 hotspots that we find. So these are basically statistical methods that find these more commonly mutated amino acid positions. Obviously, you don't need any statistics to find the most common ones. So these are the first 6 most common ones, which are well known. Some of them have tissue-specific distributions, but they've all been validated in the literature. Those are very common and easy to detect, and nobody needs statistics. It gets a little trickier, and I'm now zooming into the y-axis here, when you look at mutations that occur 2, 3, 4, 5 times, depending on the exact nucleotide context and the background mutation rate in the given tumor. Some of those are actually more likely to happen by chance, and others are actually extremely rare to happen by chance. And so you can get hotspots that are only found in about 5 samples, like this one here. And if you add even more tumors eventually, and we just showed that this is the case, this S335C hotspot in ERB2 that we here found in 2 non-small cell lung cancer samples is actually beginning to become a statistically significant recurrent hotspot. It also tends to occur in tumor samples that have no other driver in the EGFR or RTK-RAS pathway in lung cancer. So even rare ones can become important. So that's one resource that we have, and we've linked that up to, we've made that available on the web, so you can go to the website here. You can look for genes or mutations. I'm typing in, this is actually an older version, so for ERB2, the first iteration found 6 hotspots, which are all listed here, and you can interact with this in more detail. ERB2 S310F was found in 26 samples, mostly S310F, mostly S310 to F, sometimes Y, and then you can get tissue distribution across different tissues as well. The second resource that we're building at MSK is one called OncoKB, and that is simply, it's one out of, there are many of these knowledge bases out there. Some of them are more private, others are more public, others are more complete than others. And the one that we're developing at OncoKB is primarily developed for our own internal use. Every patient who gets their tumor sequence at MSK gets a report of all the variants with annotations from OncoKB. And the idea is to annotate basically the driver variants found in a tumor and then identify those that are potentially actionable. We have made the vast majority of the data publicly available at onkokb.org for research purposes. And you can see we've now worked our way through 519 genes. We have annotations on over 4,000 alterations, 64 tumor types, 76 drugs. And at the heart of it, once you get to the clinical actionability, is a level of evidence system that we've established is similar to those used by many others, where we basically distinguish for a given variant in a given tumor type the level of therapeutic implications, where a level 1 alteration is an FDA-approved biomarker predictive of response in a particular indication. So V600E in melanoma is a prime example. Level 2A is mostly those variants that are NCCN guideline listed, but not quite yet or may never be an FDA-recognized biomarker. Level 3 are those, and this is for anybody in clinical trials, level 3 and 4 are those where there isn't an FDA-recognized FDA recognition yet, but these are variants for which there's compelling evidence either already in clinical trials or in clinical studies, that's level 3A, or preclinical studies or interesting case reports, anecdotes, those are level 4. So those are, for example, ERB2 mutations, AKT1 mutations in breast cancer, or IDH1 mutations in several cancer types. And those are the ones that we are particularly interested at MSK in catching so that patients with these mutations can be enrolled in appropriate clinical trials if they're available, or some other kind of treatment if available. Just a quick overview. You can go to the website at onkokb.org to see all the genes. So currently, we have 17 genes in the level 1 bucket, so FDA-approved indications. Level 2, 10 genes, 26 in level 3 and 13 in level 4. This is a moving field, so we, depending on the latest literature or like meetings like ASCO or ACR, we quickly move things around here and promote them from 4 to 3 or vice versa as needed. And this is just an application of the data across different cancer types. So just simply asking what fraction of tumor types across patients actually have actionable or potentially actionable alterations. And GIST tumors are, 75% of GIST tumors have actionable alterations, so these are mostly level 1, KIT mutations mostly. Breast cancer stands out here in that it has a very high number of what we call level 3 mutations, so potentially actionable, but not yet FDA recognized. So about 50% of those are level 3, and then what you see here at the bottom, the 10% or so, those are, or 15%, those are the HER2 amplifications that are obviously level 1. But then on the right, this tail here of this graph has lots of tumor types, which, yeah, we may be able to find driver mutations, but even there are very few potentially actionable variants found in these cancers. And here's just an example of what our reports look like. So we decided to put less on there. Less is more. So this is a lung adenocarcinoma case in which we found an EGFR in-frame deletion in exon 19, as well as an ARAF missense mutation at amino acid 588. In this case, the indel in EGFR is a known driver, so it gets the OncoKB bullseye, and it's a level 1 alteration, sensitizing to erlotinib, afatinib, gefitinib with and we get this, put this little text in here. The ARAF variant, on the other hand, even though this is a known driver gene, a cancer gene, it's a variant of unknown significance, so it does not get an annotation here. And how do we put all this together? So we've, for the last 10 years now, I've been developing the cBioPortal for cancer genomics, and thereby made it very easy for researchers and clinicians to access the data, but we also made it very easy for them to misinterpret the data, which is why we, over the last few years, added these links basically to cancer hotspots, to OncoKB, to other databases as well. So we hope to make it easy, but not too easy, or not too easy to make mistakes. So the challenge was always, obviously, I mentioned this, data in Excel spreadsheets is not a good way for anybody to analyze data. So we put all of that data into a database. This was 10 years ago with the first TCGA project, and over time we've evolved this tool into, I think, a useful tool, trying to always keep it intuitive, try to keep the response times quick, which is difficult as we're now gathering more and more data, and we have data sets of 40,000 in AACR Gini, for example, so that makes this very hard. One thing that maybe everybody out here should know is, so we've released the software under an open source license, meaning anybody can download the software, install it locally. Many institutions do this. So, for example, Dana-Farber has an instance running with all their tumor samples. Princess Margaret, CHOP, Cornell, the Hive, we're actually all collaborating together on the software. All of us have local instances, but there are many more. The company called the Hive that helps us connect with pharma companies. So lots of pharmaceutical companies now have instances running. They actually help us develop features. All of the features that they build go back into the main software branch and are therefore available to anybody. Just some statistics on the use over the years, but I'll skip that. A couple of other instances. So AACR Project Gini has one instance where you can access all of the Gini data, and then we maintain lots of local instances, private instances for Stand Up to Cancer projects, or the Poetic Trial Consortium, TCGA, many more. And then this is what this looks like at MSK now, where we have all of the data that is generated at MSK, be it in the clinical sequencing space, where we now have sequenced about 30,000 patients, or be it in the research space. All of that is automatically inserted into cBioPortal, where it lives alongside the publicly available data, TCGA, 40,000 samples curated from the literature. It's integrated with OncoKB, with other information about recurrence, And then our newest challenge, obviously, is getting the clinical data in there as well. And I have one example at the very end that shows you what that could look like if it's done right. All of that is access controlled. Anybody at MSK actually has access to all of the clinical sequencing data, and there's a publication committee that then governs the use of that data. But the research data is private to those who requested the samples to be sequenced. And I think I have a few minutes left to show a couple of examples. cBioPortal is, I think, relatively easy to use. If you know someone who uses, you can learn a lot just by looking over that person's shoulder. So this is an example of the published MSK impact data set. This is found in the public portal at cBioPortal.org. It has 10,000 patient samples in there now. You can see 1,600 lung cancers, 1,300 breast, lots of other cancer types. If you scroll down the list, you can always search these tables over here. And by default, it shows you the mutation frequencies across the entire cohort. So in this case, TP53 is the most frequently mutated gene at 40%, followed by KRAS, TERT, and then over here you can see copy number changes. So the CDKN2A B locus is the most frequently deleted one at 8% or 7%. MYC and cyclin D1 are most frequently amplified. Down here you can see male-female sex distribution, sample types at the bottom here. We have about 60% primaries, 40% mets. And then you can make subselection. Anytime you make a subselection, all graphs update. And here I'm selecting the non-small cell lung cancers. Then within that, the lung adenos. I'm now down to 1,300 samples. And you can see all of the gene lists have updated now. P53 is now at 50% in that subset, followed by KRAS, EGFR. So you can very quickly assess frequencies and cancer types. You can then dive deeper and say you want to know more about genes. So in this example, I'm picking genes in the receptor tyrosine kinase RAS pathway. So you can see I've now picked 5. Those 5 are then popped up there in this little query field. I'm now manually entering a couple other genes that I'm interested in the same pathway. So you can see what I'm doing here: EGFR, receptor tyrosine kinases, RAS, RAF genes, and then I hit query. And when you do that, you get this view, which now tells you, so I'm now zooming into this cohort and I want to see more details. So I've now selected 1,300 samples, lung adenocarcinoma samples. This basically is a giant spreadsheet in graphical representation. So each column, there are 1,300 columns here. Each column is a patient. Each row is one of the genes that you've queried. And then the graphical representation shows you how that gene is altered. And everything is sorted by the first gene down to the last gene. So EGFR is altered in 30% of cases here. And then genes at the bottom here, BRAF is 7%. You can change the sort order if you want. And then the alterations are shown in those little graphical representations. You can see at the very right, there's about 10% of cases of 14 that have no alterations. EGFR is altered by amplifications. Those are the tall rectangles, or by in-frame insertions, deletions, or missense mutations shown in red and then green. Red, ROS, and ALK, the purple bars here indicate fusions. So those are the genes that are commonly altered by fusions. And then going back to this driver passenger, you can see the light green ones here at the end. I'm just going to pause this real quick. Those are mutations of unknown significance. You can see a couple of them in EGFR. You can see a few more relatively in RET and ROS, and then again in ALK. Those are the ones that are potentially passengers, and those are obviously more common in RET, ROS, and ALK, which are actually not expressed unless they're activated by a fusion. You can change all the settings here, change mutation colors. You can bring in clinical tracks. I'm only going to show you total mutation count here now for each sample, as well as the mutation spectrum, which is a composition of all the nucleotide changes found. And you can see little things, for example, the fusions occur in tumor samples these are all lung adenocarcinomas, remember, in tumor samples with low mutation burden. But the VUSs in these genes occur in tumors with high mutation burden, again, suggesting that they're likely passengers and not contributing to the tumor growth. So you can begin to see here the color coding of the mutations is linked to OncoKB. It's linked to hotspots, so any possible driver is marked as such. And like I showed you earlier, you can change your settings here. And now what I'm doing is I'm actually hiding the VUSs. And you can see this pattern of mutual exclusivity, which you would expect across a cohort of lung adenocarcinoma cases, is getting much better. So all of those contaminating passengers are now gone. And you can zoom in further. You can look at lollipop plots for individual genes. So all of the plots I showed earlier, you can make here on the fly I'm going back to ERB2 because we looked at that earlier. You can quickly see the mutations. You can see a couple of mutations in the N-terminal domain, and then the majority of mutations are in the kinase domain. You can see summary statistics. The table below shows you details, and this is where, again, the annotation comes in from OncoKB, from cancer hotspots. So it's all relatively easy to use, and with a little bit of use, you actually get the hang of it quite quickly. Here's an example of my cancer genome, which we're pulling in, as well as the civic database. You can get down, and if you show more, you can get to the very end where you can now see some variants that actually have no annotation here. So these are potential passenger variants in lung adenocarcinoma case. And then last, I think I'm out of time, I'll just show one quick glance at a slide. If you now want to zoom in all the way into a patient, and this is actually a patient sample, a glioma patient from MSK, where we now automatically feed that data in daily. This is a patient, 38 months survival, recurred 18 months after diagnosis, who had an original diagnosis here, that tumor sample number 1, was on a clinical trial. We can't show the drug name here, but investigational therapy here. Looking from left to right is a timeline, and then the patient progressed about 18 months after diagnosis. Tissue was biopsied at 18.5 months, and then again at 22 months. And what you're now comparing is genomic differences between the tumor samples. What you're looking at here at the top is copy number profiles. You can see by eye the EGFR amplification on chromosome 7. That's listed down here. So all 3 tumor samples have the EGFR amplification. But then most notably in this case is you went from 3 mutations to over 100 mutations over the span of 18 months of treatment for this patient. TERT, EGFR, PIK3R2 are in common in all 3 samples, but then samples 2 and 3 have over 100 additional mutations, probably induced by, it's probably an alkylating agent, temozolomide or something, that induced all of these extra mutations. And some of them you can see here, when you sort the column the right way, you can see are actually known driver mutations that now this patient has in the tumor after treatment. And I hope that gave you a good idea of what you can do with the cBioPortal, that if the right data is in there, you can do a lot with it. So in summary, to make sense of variants identified in tumor samples, we can use simple tools like recurrence to identify frequently mutated genes, not just genes, but most importantly, the amino acids in the genes. We can use prior knowledge, like our OncoKB database, and then you can bring all that together in a tool like cBioPortal, and then you hopefully have the tools that you need to identify the relevant mutations in a tumor sample. And with that, I just want to acknowledge all the people who are behind the work and can take questions if we have time.